Hi everybody, welcome back. Today we're going to be talking about membrane structure and transport. The plasma membrane has three main functions. The first is it defines the borders of the cell. What this means is that anything inside the plasma membrane is inside the cell, and anything outside the plasma membrane is outside the cell. Plasma membrane is also selectively permeable. And what this means is that some things can go right through the plasma membrane and other things cannot. They have to use other mechanisms to go through. The third function for the plasma membrane is that it helps to manage communication with other cells using receptors and markers on the cell surface. The membrane has many different kinds of molecules within it. So the majority of the plasma membrane is made up of phospholipids. However, it's also associated with different proteins, carbohydrates, and cholesterol. The phospholipid bilayer is going to spontaneously form into this bilayer because of that amphipathic nature of the phospholipids. And because the aqueous environment of the body, the hydrophobic tails don't want to be near the water, so they spontaneously try to hide from it, and you end up with the hydrophilic heads on the outside of the cell and the hydrophilic lipid tails packed together, and then another hydrophobic head on the inside of the plasma membrane. The plasma membrane is also associated with various proteins. They have several functions. The first will be to transport substances across the membrane when the substance can't go through the membrane. The second will be to function in enzymatic reactions. The third is they help to a role in recognizing self from non-self in various types of communication. The fourth is that they help to form cellular junctions to pull tissues together. And the last one is they provide structural support for the cell. There are two different categories of membrane proteins, and it depends on where they are in the plasma membrane. Integral membrane proteins are associated directly with that hydrophobic core, where the hydrophobic tails are. Two types of proteins. One is a transmembrane protein. This is a protein that goes all the way through from the extracellular side to the intracellular side. And sometimes they can go across several times, and we call those multipass proteins. The other type of integral membrane protein is a lipid-anchored protein. And this is a protein that may not go all the way across, but it is associated with that hydrophobic core. The other type of protein is a peripheral membrane protein, and these do not directly touch that hydrophobic core, but are instead bound to other membrane proteins. The plasma membrane is also associated with different types of carbohydrates and cholesterol. When an oligosaccharide is associated with a protein within the plasma membrane, we call it a glycoprotein. And we use glycoproteins on our cells to designate self from non-self. When you have an oligosaccharide that is associated with a phospholipid or bound to a phospholipid, we call it a glycolipid. We also have cholesterol within the plasma membrane. It's composed of 4 fused rings, and it's going to fit into one side of the bilayer. The plasma membrane is not static. It's not just sitting there. It's constantly moving around and changing. The phospholipids themselves are in constant motion. They can rotate around, so spin around. They can move laterally throughout the plasma membrane, and they can also flex those fatty acid tails. However, moving from one side of the lipid bilayer to the other side is very rare, and in fact is only done when you have special transport proteins to move those phospholipids. Membrane proteins can also move laterally through the plasma membrane. However, because they're so much bigger, they move a lot slower than phospholipids. So you end up with a model of the plasma membrane where you have many, many phospholipids with the fatty acid tails on the inside and the hydrophilic heads either on the extracellular side or the intracellular side. You have integral membrane proteins going all the way through the plasma membrane. They can be associated with either peripheral membrane proteins or glycoproteins. And you also have glycolipids, which are attached to the phospholipids. Now, the fluidity of the membrane can change. Things can move faster or slower through the membrane. 3 factors that affect the membrane fluidity. The first one is the proportion of the unsaturated fatty acids. And we talked about this when we talked about macromolecules. When you have unsaturated tails, they have a double bond and they have a kink in them. That kink allows the phospholipids to not be quite as close to one another, which means there's basically just more room for them to move through the bilayer. However, when you have lots of saturated tails, the phospholipids tend to pack very, very, very closely together and make that plasma membrane much more rigid and viscous. The second factor that affects membrane fluidity is temperature. As the temperature increases, the membrane becomes more fluid. Cholesterol is the third factor that's going to affect membrane fluidity. Cholesterol is going to pack in and fit in the spaces in between the phospholipids that are created by the kinked tails. Cholesterol's job is to help resist changes to the membrane fluidity. At our body temperature, cholesterol mainly reduces the fluidity of the membrane, so it's going to make it more rigid. Cholesterol at low temperature would help to keep your plasma membrane from becoming too solidified. And at very, very, very high temperatures, it would keep your plasma membrane from melting and falling apart. When we talk about moving things across the membrane, we have to first talk about the concentration gradient. So concentration refers to how much stuff is dissolved in a liquid. The fancy way of saying stuff is solute. The fancy way of saying liquid is solvent. Concentration is the amount of solute in a small volume of solvent. If you look at the picture below, imagine that each blue dot is a molecule of solute. On the right-hand side, you have lots of it, a very high concentration of solute. However, on the left-hand side, you have a very low concentration of solute, not very many blue dots at all. This gives you a concentration gradient, a change in the concentration. When molecules move down the concentration gradient, they are moving from an area of high concentration to an area of low concentration. It is easier, meaning it takes less energy, to move a molecule down its concentration gradient than to go up the concentration gradient. We use concentration gradients to move stuff into and out of cells all the time. They come in and out of the cell via membrane transport. There are two types of transport that are classified depending on which direction the concentration gradient is going. So in passive transport, you're moving from a high concentration of solute to a low concentration of solute. You don't need any cellular energy for this. And there are three types of passive transport: fusion, osmosis, and facilitated diffusion. The other type of transport is active transport. And this is going the opposite way. You're going from a low concentration to a high concentration. So you're going up the concentration gradient. This does require cellular energy. And there are a couple of different types of active transport processes. One is active transport pumps, and the other one is bulk transport. Exocytosis, where you're moving things out of the cell, or endocytosis, where you're moving things into the cell. Let's start with diffusion. This is passive transport. So we're moving a substance from an area of high concentration down its concentration gradient to an area of low concentration. So if you look at the picture, you can see there's a bunch of yellow molecules and a membrane. The yellow molecules are at a high concentration on the left side of the membrane and at a low concentration on the right side of the membrane. And they will move down their concentration gradient from the area of high concentration to the area of low concentration. This is diffusion. Once they get to be equal on both sides of the membrane, they're not going to stop moving. They will continue to move back and forth across the membrane, but there's going to be no net movement. You can also have diffusion with more than one solute. So if you look at the bottom, you have yellow balls on the left side of the membrane at high concentration, and you have purple balls on the right side of the membrane at high concentration. And their concentration gradients are in the opposite direction. So the yellow balls will move to the right, and the purple balls will move to the left. They're both moving down their concentration gradients. Neither one of them requires any energy to do this, and so this is passive transport. And they will continue to do it until they equal out on both sides of the membrane. This example that we're looking at is a permeable membrane. It is permeable to both the yellow and the purple balls. But what happens when you have a plasma membrane, which is semi-permeable? Some things can go across, some things can't. So the things that are small, hydrophobic, and uncharged like oxygen and CO2 and steroids, can move across the membrane relatively quickly. Remember, the inside of the membrane is hydrophobic, and so hydrophobic molecules are perfectly happy going through there. If you have a polar molecule that's uncharged, that's small enough, it can also move across the membrane. However, not quite as easily as hydrophobic molecules. Examples of small uncharged polar molecules would be water, ethanol, urea. However, if they get too big, if you have large uncharged polar molecules like glucose or sucrose, they're not going to be able to move across the membrane by themselves. They're going to require something else to bring them across. And the least likely thing to go across the membrane is anything that's polar and charged, like ions. They're not going to be able to move across the membrane by themselves and are going to require some kind of transport protein. Remember that ions are hydrophilic. They're charged and they're polar, so they're going to dislike that hydrophobic core of the membrane. Osmosis is just a special type of diffusion in which we're talking about the movement of water. So this is still passive transport. Imagine you have a tube in which there's a membrane that's permeable to water, but not to the solute. Stop the video and count the green balls of solute on each side of the membrane. There are 6 balls on the left side of the membrane and 13 on the right. So we have a concentration gradient because there's a difference in the concentration of the solute on one side of the membrane and the other. However, this membrane is not permeable to the solute. The solute can't move across the membrane to even out, but the water can. The water is going to move towards the side of the membrane where the most stuff is. The concentration will end up being the same on each side of the membrane. We haven't moved the stuff, but we have changed the volume. We've reduced it on the left side, and we've increased it on the right. The key part to this is to figure out which way will the water move. And I'll tell you how to figure that out. The water always follows the stuff. So when you have a lot of stuff, a lot of solute, and the stuff can't go across the membrane, the water will go across the membrane. Well, how do you know where most of the stuff is? We use the tonicity of a solution to tell us where the stuff is. So the tonicity of a solution is just a comparison of 2 solutions on either side of a membrane. If the 2 solutions are isotonic, it means they have the same amount of solute on each side. However, if the concentration of the 2 solutions on either side of the membrane is different, then one is going to have more stuff and the other one will have less stuff. So the one that has more stuff is called hypertonic. That has the higher concentration, whereas the one with less stuff is called hypotonic. That's the one with a relative lower concentration. Water will always move from the hypotonic solution towards the hypertonic solution because it will be following the stuff. The last type of passive transport we're going to talk about is facilitated diffusion. And the difference here is solute isn't going directly through the membrane. It can't fit. Maybe it's too big, maybe it's charged. And so you have to have a membrane protein involved. There's 2 types of those. One is a channel protein. This is going to open and close and allow small hydrophilic molecules to go through, things like water or ion. You can also have a carrier protein, and this, instead of being a channel that just simply opens or closes, a carrier changes the conformation and actually opens on one side of the membrane and then sort of swivels and opens on the other side of the membrane. So it'll carry larger molecules across that membrane. But again, it's passive transport because we're going from a high concentration to a low concentration of solute. Active transport involves the use of pumps, and this requires energy. And it's going to transport a substance, but in the other direction. So it's going to go against the concentration gradient. In active transport, you're going from a low concentration to a high concentration bulk transport. And this is using vesicles or membranous sacs. Exocytosis is moving something out of the cell, and endocytosis is moving something into the cell. We already talked about active transport and exocytosis when we talked about the endomembrane system. Endocytosis has 3 different types of bringing in molecules from the outside. The first type is phagocytosis, and that's going to be bringing in particles from outside the cell. Pinocytosis is bringing in small sips of fluid from the extracellular area into the cell, and receptor-mediated endocytosis is stimulated by the binding of special receptors on the cell surface. All three of these types of endocytosis, as well as exocytosis, require energy because we're moving things against their concentration gradient. That's it for today. See you in class.